Clinical chemistry, chromatography, mass spectrometry, and enzyme and rate analysis. Chromatography is a group of techniques used to separate complex mixtures based on different physical interactions. The basic components of chromatography include a mobile phase, which is gas or liquid that carries the mixture or the sample. The stationary phase, which is a liquid or solid that interacts with the sample. A column holding the stationary phase. And then the separated components, or the eluate. Similar to gel electrophoresis, when we talked about gel electrophoresis, we talked about how the gel itself had different sized pores in it, and then the larger molecules would move through, such as DNA or proteins, and they would migrate through based on their charge and their size. Kind of a similar concept, except instead of stopping in the middle of the gel and us evaluating those larger compounds, we're talking about very small molecules, gases, and they're going straight through the column and separating out as eluate in the end. There are four major types of chromatography: adsorption, partition, steric exclusion, and ion exchange. In adsorption chromatography, it's also known as liquid-solid chromatography. The competition between the sample and the mobile phase for adsorption sites on the solid leads to that separation. More soluble substances are going to move faster than less soluble. The mixture is typically separated by polar functional groups. The stationary phase can be acidic polar, so a silica gel, basic polar, and alumina, or nonpolar, such as charcoal. The mobile phase can either contain one solvent or multiple solvents. This is not common in clinical chemistry. Partition chromatography is also known as liquid-liquid chromatography because the stationary phase is a liquid instead of a solid, as you would see in adsorption chromatography. The separation of solute by relative solubility in an organic or nonpolar solvent and an aqueous polar solvent. In other words, the relative solubility between the stationary and mobile phases leads to the separation. It's performed in a separatory funnel. Molecules with polar and nonpolar groups in aqueous solution are added to an immiscible organic solvent. It's then shaken vigorously and then allowed to separate. Polar molecules remain in aqueous solvent, while nonpolar molecules are extracted in the organic solvent. This works best with non-ionic molecules. In steric exclusion chromatography, it's also known as liquid-solid chromatography, it separates solute molecules on the basis of size and shape. Chromatographic column is packed with porous material. Samples with different sized molecules are moved down the column and dissolved in the mobile solvent. Smaller molecules enter the pores in that packaging material, and they get trapped momentarily, while larger molecules move more quickly through the column. Packing material can be purchased with different size pores depending on the molecules that need to be separated. So this is a picture showing those packing materials with the pores in them. You can change the pore size depending on which molecules you want to separate. Smaller molecules are going to get trapped and held up in there momentarily, while larger molecules are going to go straight through. And then you can see you can have separation. In ion exchange chromatography, the solute mixtures are separated by the magnitude and charge of ionic species. The stationary phase is a resin that consists of large polymers with charged functional groups. The resin is insoluble in water, and functional groups are immobilized as side chains on resin beads that fill a column. There's cation exchange and anion exchange ion exchange chromatography. So in cation exchange resin, hydrogen ions are released and cations such as sodium ions are captured. So the hydrogen ions that are on the surface of that resin are released and exchanged for the cation. What comes through is the hydrogen ions, and then you can release the cations later by adding an acidic solution. In anion exchange resin, hydroxyl ions are exchanged for anions. So what comes through the column is the hydroxyl ions, and then the anions are trapped in there, so they exchange places with the hydroxyl ions. In mixed bed resin, anion and cation resins are mixed together and used to deionize water. Displaced protons and hydroxyl ions combine to form water. Ion exchange chromatography, in general, is used to remove interfering substances from a solution, to concentrate dilute ionic solutions, and to separate mixtures of amino acids. So this is a picture of the negatively charged functional groups on the resin, and when we put the sample through, you can see what's coming through to the end. Thin layer chromatography. Thin layer chromatography is a variation of column chromatography. There is a thin layer of sorbent, such as alumina, silica gel, cellulose, or cross-linked dextran, which is coated on a glass or plastic plate. Each sample is applied toward the edge of the plate. One edge of the plate is placed in solvent, which migrates up the thin layer by capillary action, dissolving and carrying sample molecules up with it. The separation occurs by any of the methods described previously, depending on what thin layer you added to the glass and the solvent that you've chosen. After, the sample components are compared to known standards on the same plate for identification. It's typically used as a semi-quantifiable drug screen test using a densitometer. RF is the ratio of the distance moved by the unknown solute to the distance moved by the solvent in the mobile phase. So the calculation is RF is equal to the distance of the leading edge of the component moves divided by the total distance the solvent front moves. For example, if the unknown migrated 9 centimeters and the solvent migrated 37 centimeters, what is the RF? The RF is 0.24. That 0.24 can then be compared to known solvents on that same plate, and then you can determine the identification of unknowns. So this is a picture of what thin layer chromatography looks like. So this plate here is put into a box. There's the solvent at the bottom of the container. You apply the samples and the standards at the bottom, and then you're just going to dip the edge into the solution. The solution is going to start to migrate up by capillary action, and then as the molecules within that solution are going to separate out, they're going to have fronts at different locations, and then we can compare the standards to the samples to see what the unknown identifications are. So you can see unknown 1 has this in it, and it also has this in it. Unknown 2 has this in it. High performance liquid chromatography, or HPLC, uses pressure, controlled temperature, inline detectors, and gradient elution techniques. So you have a pump which pumps through the column, and then there's a detector, waste is released, and then there's a display that happens on a computer. The pump forces the mobile phase through the column faster than what gravity can. A column is the stationary phase that's packed into long columns, usually kept at room temperature, but it can be heated to increase the rate of partition. Fine, uniform packing creates more peaks, but requires more pressure. The most common packing material is silica gel. The detectors monitor the eluate as it leaves the column and produce an electronic signal proportional to the concentration of each separated component. Unknowns can be compared to a library of spectra of known standards, and then concentrations are calculated as areas under the peak. So as we see spectra, it'll be a line, and then you'll see some peaks when different substances are going to emerge. The peak is known as a spectra, and that's compared to a library of different spectra, and then the area underneath that peak can be calculated as the concentration of that substance, as long as you controlled for like the concentration you inserted a known concentration of standards, we can do the calculation. Gas chromatography is used to separate mixtures of compounds that are volatile or can be made volatile, and it uses boiling point. So volatile means that it can be turned into a gas or can evaporate. GC can be a gas solid, where you have a solid stationary phase, or gas liquid, non-volatile liquid stationary phase. GLC is more common in the clinical lab. It's similar to HPLC, but the mobile phase is a gas, such as nitrogen, helium, or argon. The sample must be injected as a gas or must be evaporated at the entry point. Volatile compounds with lower boiling points are going to move more quickly through the column. A chromatogram is used to identify compounds by their retention time, and they can determine concentration by the area under the peak, similar to how you can do that with HPLC. Mass spectrometry is used as a detector to identify samples that are eluting from gas chromatographic or HPLC columns. So we have GCMS, so gas chromatography mass spectrometry, or LCMS, liquid chromatography mass spectrometry. And they have powerful analytical capabilities with widespread clinical applications. When the sample is introduced, it has to be ionized. And there's two major methods of ionization: electron ionization or atmospheric pressure ionization. Electron is the most common for GC mass spec. High energy electrons bombard the molecules, which causes them to form charged molecular ions and fragments. The molecules break down into characteristic fragments according to their molecular structure. Qualitative identification is based on the comparison to reference spectra. In atmospheric pressure ionization, it's more common for LCMS. Electrospray ionization and atmospheric pressure chemical ionization are examples of this. So the sample is volatilized or evaporated and then ionized to form a charged molecule. Molecular ions and fragments are separated according to their mass to charge ratio, or MZ ratio. The detector gives the intensity of the ion current for each species. The mass analyzer can be a quadrupole, which is the most common mass analyzer used today, an ion trap, which is a modified quadrupole, MS-MS tandem, which is the use of mass spectrometers for greater selectivity and lower detection limits, or high resolution MS. The detector, the use of an electron multiplier to detect ions. Applications of MS in the clinical lab. MS is used for structural and molecular weight determinations, small molecule analysis, such as drugs of abuse and urine toxicology. Proteomics. So proteomics is different from genomics. Genomics investigates the composition of DNA and looks for different genes that are present in the DNA. But as we know, sometimes those genes are not actually turned into protein. So the genes exist, but they're not actually functional. They're not transcribed and translated into protein. If the genes are present, it doesn't mean that there's going to cause a disease or that whatever that gene is associated with is actually going to produce that protein. So it's more important to look at what proteins are actually present. So in proteomics, it's the investigation of protein products that's encoded by the genes. And as I mentioned, it's more important than genomics for disease detection. Two-dimensional electrophoresis. So the first dimension, proteins are resolved according to their isoelectric points using immobilized pH gradients. And then the second dimension, proteins are separated according to The MALDI-TOF and CELDI-TOF mass spectrometry is used for the analysis of biomolecules such as peptides and proteins. And MALDI is also used for pathogen identification. So isolated bacterial or fungal colonies can be directly spotted onto the MALDI plate and ionized, which results in a protein fingerprint of the species. Significantly cheaper and faster than traditional automated biochemical identification techniques. So this is a picture of the spectra for the MALDI of different species. So if we have an unknown species, we can compare it to the library of known spectra. There is one other technique that I just wanted to mention here, and that's enzyme rate analysis. Measuring the activity or mass of enzymes that are predominantly intracellular and present in low concentrations is important. The rate of an enzyme catalyzed reaction is directly proportional to the amount of active enzyme in the sample. Rates are measured by the time in decreasing the concentration of a substrate under controlled conditions. Measurement of substrates is performed using spectrophotometry, fluorometry, or chemiluminescence. So basically, to determine if an enzyme is active and determine the rate of that enzyme, we determine the disappearance of the substrate or the formation of products. And the disappearance of substrate or formation of products can be evaluated using other techniques that were discussed previously.